Genetics. The genes GYPA and GYPB, which code for GPA and GPB respectively, are located on the chromosome 4 at 4q, 28q, and 31. The known alleles for GYPA is MN, and for GYPB is S, are codominant. The genes are highly homologous, meaning they are very similar and probably arose from gene duplication. GYPA is considered to be the ancestral gene, and GYPB is organized into 7 exons. GYPB has a size of arrangement similar to those of GYPA, but has only 5 coding exons plus 1 non-coding or pseudoexon. Misalignment of the GYPA and GYPB during meiosis, followed by an unequal crossing over, appears to explain some of the variant glycophorins observed in the MNS system. So the point of the fusion between GPA and GPB part in the hybrid glycophorin can give rise to novel antigens. Gene conversion involves a non-reciprocal exchange of genetic materials from one gene to another homologous gene. So as with hybrid resulting from crossing over, the amino acid sequence at the junction of the hybrid glycophorins can give rise to novel antigens of low prevalence. GPA and GPB deficient phenotypes. So RBCs of 3 rare phenotypes lacks GPA and GPB, or both GPA and GPB. Consequently, they lack all MNS antigens that are normally expressed on those structures. So for the U phenotype, the U antigen is located on GPB, very close to the RBC membrane between amino acid 33 and 39. The RBC is usually type SSU, and these individuals can make antigen U in response to transfusion or pregnancy. Anti-U is typically IgG, immunoglobulin G, and has been reported to cause severe and fatal HTRs and hemolytic disease of the fetal newborn. The U antigen is resistant to enzyme treatment. So most example of anti-U react equally well with untreated and enzyme-treated RBCs. However, there are rare examples of broadly reactive anti-U that do not react with papain-treated RBCs. There is also a strong correlation between the low prevalence antigen HE, found in about 3% of African Americans, and UVAR expressions. Because examples of anti-U are heterogeneous, U negative units selected for transfusion must be cross-matched to determine compatibility. Many examples of anti-U are actually anti-U plus anti-GPB. If the patient is U negative and N negative, the antibody may actually be potent anti-N plus anti-U, making the search for compatible blood more difficult. Negative phenotype. So in 1969, Darnborough and co-workers and Helm and colleagues 29 describe an antibody to the same high prevalence antigen called ENA for envelope, which reacted with all RBCs except those of the proposite. Most ENA negative individuals produce anti-ENA, which is an umbrella term for reactivity against various portions of GPA, unrelated to M or N, but not all antibodies detect the same portion. Anti-ENATs recognize a trypsin-sensitive area on GPA between amino acid 20 and 39. Anti-ENAFS reacts within a fisin-sensitive area between amino acid 46 and 56. And anti-ENAFR reacts with a fusin-resistant area around amino acid 62 to 72. Although the genes responsible for this phenotype has been termed EN, it's known that the ENA negative phenotype has more than one origin. Most often, the ENA phenotype results from homozygosity for a rare gene deletion of the GYPA locus. Consequently, the GPA is produced but GPB is not affected. This type of ENA negative inheritance is called ENA negative Finn, representing the type described in the Finnish report. Anti-ENA can be confused with two other specificities that react with all normal RBCs, anti-PR and anti-WRB. Anti-PR does not react with enzymes-treated RBCs and can be confused with anti-ENAFS. Anti-WRB does not react with ENA negative RBCs but does react with enzymes treated RBCs and can be confused with anti-ENAFR. Anti-ENA has caused severe HTRs and HDFN. MK phenotype. The rare silent MK was named by Metaxas and Metaxas Bowler in 1964 when they found an allele that did not produce M or N antigen. So the MK phenotype does not have an M or N antigen. A second family showed that MK was also silent at the SS locus. Several other MK are heterozygous and have been found in 1979. Two related MK blood donors were found in Japan also. The RBCs of these individual types MNS as U ENA are negative, but they had a normal hematologic picture. More individuals have been identified, and it's known that the MK gene represents a single near completed deletion of both GYPA and GYPB7. So the MK gene is a representation of the deleted gene GYPA and GYPB. Thus, MK is the null phenotype in the MNS system. The MK genotype is associated with decreased RBCs, sialic acid content, but increased glycosylation of RBC membrane bands 3 and 4.1. So other antibodies in the MNS system are antibodies to the antigen are rarely encountered and can usually be grouped into two categories. So those direct against low prevalence antigens and those directed against high prevalence antigens. Antibodies to high prevalence antigens are easily detected with antibody detection RBCs. But antibodies with low prevalence antigens are rarely detected by the antibody detection tests that are seen in as an unexpected incompatible cross-match or an unexplained case of the HDFN. Other antibodies when the antibodies to low prevalence antigens are encountered, it is common practice to transfuse units that are cross-matched compatible at 37 degrees Celsius and in the antiglobulin phase. Typing sera from MNS antigen other than M and S are not generally available, so the antigen status of compatible RBCs can be seldom confirmed. If the antibody is detected to a high prevalence antigen, the assistant of an immunohematology reference laboratory is generally needed to identify the antibody and obtain appropriate antigen negative units. So for the disease association, the GPAM may serve as the receptor by which certain pyelonephritogenic strain of E. coli gain entry to the urinary tract. The malaria parasite Plasmodium falciparum appears to use alternative receptors including GPA and GPB for cell invasion. Some of these receptors also involve NANAC.